Testing, 1, 2, 3. Everything looks good here. Hello and welcome. Today we'll be talking about lipid absorption and digestion.
Ooh, what are lipids?
Good question. Lipids are a crucial part of the cellular membrane. They help also store energy in the body.
Oh, what are lipids?
Well, I guess our friend Daniel here will help.
Ooh, who's Daniel?
Alright, today we will be going over some topics today on lipids. We'll be going over the basic characteristics, the functions in the body, lipid digestion, lipid absorption, and lipid metabolism. Some of the basic characteristics of lipids are they're hydrophobic. Their most common lipid is triglycerides, which are made up of 1 glycerol and 3 fatty acids. Each glycerol yields 16 ATP. Each fatty acid yields 120 ATP each. Some of the basic functions of these triglycerides in the body are for insulation in the form of heat, absorption of fat-soluble vitamins such as vitamins A, D, E, and K, and also it's used as a source of fuel. Another function is that it cushions the organs on the inside and the myelin sheath in the brain and nervous system.
Thank you, Daniel. I'll take it from here. So, when we eat, we chew our food. This process is called mastication. It occurs in the oral cavity. Lipid digestion actually occurs as well in the oral cavity. It is the start. Lingual lipase begins the chemical breakdown of lipids. After we finish masticating our food, it travels down the esophagus and into the stomach. This is where gastric lipase comes in handy. It assists as well in breaking down the lipids. Oh, but nothing compares to the small intestines, the duodenum to be exact. The duodenum is the primary source of lipid digestion, right here. Once lipids enter the small intestines, or duodenum, they signal a chemical reaction. Chemicals in the duodenum, called entergastrones, are affected. The first gastrointron is cholecytokinin, as seen right here. They affect the gallbladder and pancreas. It tells the gallbladder, as drawn right here, to release the bile into the common bile duct and into the duodenum. It also tells the pancreas to release its pancreatic lipase into the duodenum. Secretin tells the liver to make more bile salts. This is a key important process for lipid digestion. Bile salts are super important.
So once this fat globule enters the duodenum, pancreatic lipase cannot break it down yet. It is still way too big. So, bile salts from our gallbladder and liver both come down the common bile duct and into the duodenum. These bile salts basically emulsify the fat globule. Emulsify just means to break it down into smaller parts. So instead of it being one globule, it breaks it down into smaller fat droplets. A unique factor with bile salts is one side is hydrophobic and the other side is hydrophilic. This really helps to attract to the fat droplets. The hydrophobic side is the side facing the fat droplet, and the hydrophilic side is the side facing the cells, epithelial cells around it, in the duodenum. This really helps with moving and mobilization of the fat droplets. However, it is pancreatic lipase that digests the fat droplets into monoglycerides and free fatty acids. So these fat droplets, called micelles, break into monoglycerides and free fatty acids. So after the free fatty acids of monoglycerides enter the cell past the mucosal layer, they go right to the endoplasmic reticulum. This is where they're resynthesized into a triglyceride. This triglyceride also picks up protein and cholesterol, now creating a chylomicron.
Hi. Once the chylomicron is formed, it goes to the Golgi apparatus. This is where it is modified for exocytosis. The chylomicron leaves the cell via exocytosis and enters a lacteal.
So this is a chylomicron.
Once this chylomicron travels through the lacteals, it enters the blood via the subclavian vein. Lipoproteins and lipase break it down for absorption. Thanks.